We will focus in this lecture on particular crop species and their evolution from the wild into the domesticated form. Today, more than 50% of modern humans' calorie input derives from carbohydrates, mostly from cereals. Without crops, we would lead different lives with different subsistence strategies, different forms of society, and different ethical values. The irreversible way into dependence on agriculture that began more than 12,000 years ago is an important evolutionary aspect in the history of humankind. Let's start with some terminological matters. Systematics investigates the relationship between organisms by involving other disciplines such as morphology, anatomy, cytology, and genetics. This results in the definition of the phylogeny of individuals. Here also paleobotany and paleontology play an important role, and in particular archaeology if domesticated species are in focus. Taxonomy is a subdiscipline of systematics with the main purpose to describe and group modern and fossil organisms according to a reasonable system. Such a system follows internationally defined rules. Under nomenclature, we understand the grouping of organisms in a hierarchical system. The important taxonomic groups are in descending order: the kingdom, the tribe, the class, the order, the family, the genus, and the species, which is the most important taxon in systematics. There are different concepts to define a species. The morphological species concept comprises the species description according to phenotypical and morphological variation. Theoretically, the distinction between two species is made where characteristics in each individual do not overlap. In reality, such clear borders do not exist. The morphological species concept has been developed by Carl von Linné, published in Systema Naturae. The Latin names of plant species go back to his nomenclature, so for example, the L after the species of einkorn, that is Triticum monococcum, is the author abbreviation for Linné. Relatively early in history, it has been recognized that this static species definition is problematic because there are smooth transitions between species. Georges Cuvier redefined the species concept and included the aspect of free assortative mating, that is, mating of similar individuals. Jean-Baptiste Lamarck advanced the view that living organisms continuously change throughout generations. He based his view on the change of domesticates under human influence and was an important precursor of evolutionary theory. With Charles Darwin, evolutionary processes coined the species concept. Today, a species is defined as a natural population in which individuals descend from each other, produce fertile offspring, and coincide in relevant characteristics. They are separated from other populations by reproductive barriers. Species can be recognized because they cannot be hybridized with others. This does not always apply, as for example with the evolution of wheat species. Many species can be further classified into subspecies. Subspecies can easily be differentiated through some characteristics. They are always geographically or temporally separated, resulting in a spatiotemporal restriction of crossing over of genes. Individuals of subspecies may have also fertile bastards if hybridized. Crop species reproduce successfully only within their own species and with their wild form. The wild and the domesticated form are two subspecies of the same species. The correct systematic species name for tetraploid weeds is Triticum turgidum. The wild form is Triticum turgidum subspecies dicocoides, which is wild emmerweed. Triticum turgidum subspecies dicoccum, domesticated emmer, and Triticum turgidum subspecies durum is macaroni or heartweed. Further differentiation of crop species is given by the varieties or cultivars, which have developed through breeding. Sometimes varieties or cultivars are also named as races. Land races, for example in wheat, have developed through genetic isolation, highly selected breed through strong selection by humans. The most important difference between wild and domesticated species is in reproduction. While wild plants distribute their seeds spontaneously, domesticated species are fully dependent on humans for reproduction. Morphologically, crop species differ from their wild relatives by the anatomical changes that go hand in hand with the reduction of seed dispersal. They also have in most cases larger and more seeds and fruits, thinner seed coats, and many of them an apical dominance, that is, the main shoot is very robust in relation to the lateral axis, a characteristic which is very prominent comparing teosinte and maize. Crops also underwent physiological changes in that they lost their seed dormancy. They generally germinate, flower, and ripen simultaneously. Also changes in photoperiod sensitivity occur, and they usually lost their bitterness in edible parts of the plants. The latter is undoubtedly a result of human selection. We heard already multiple times about the distinction of cultivation and domestication, but once more again you can read it here. This list provides an overview on the different cereal species that have their origin in the Fertile Crescent. Here you see an example for the taxonomic groups of some cereal species. Starting from the genus level, you see that the genera with domesticated species of rye, barley, and wheat all belong to the tribe of the Triticeae, the subfamily of the Pooideae, the family of the Poaceae, and fall into the class of the Monocotyledonae. You may recall the features of identification from Lecture 4, but here just to pinpoint the most important morphological differences between wild and domesticated cereals. Since the altered reproduction in domesticated species is related to a change in anatomy, archaeobotanists can make use of the features to differentiate between the wild and the domesticated forms of a species. The left photo shows two domesticated emmer ears, the photo on the right wild emmer ears that fall apart at the point of maturity. In the wild form, this leads to reproduction as the grains fall onto the ground and germinate. The stable domesticated ear can only be separated by threshing. The particular area which determines whether the ear shatters or remains intact is the attachment scar of the spikelets. In the wild form, the attachment scar is smooth, as a result of the natural release of the spikelet. In the domesticated form, the attached spikelet is mechanically broken, leaving a cultrate area on the scar. The use of chaff remains for identification has only been fully recognized during the last 10 years. In earlier publications, differentiation between wild and domesticated species was based on grains, which is not distinctive on the subspecies level. Although in theory wild chaff remains can be differentiated from domesticated chaff, preservation is often insufficient to apply these characteristics. The table shows the large amount of unidentifiable chaff remains. Destruction may be syn or post-depositional, but very often also related to processing of the cereals, that is, dehusking by ancient farmers. With wheat, more than 90% are not identifiable as wild or domesticated, because the spikelet often shows a torn attachment area, which you can see in picture B. Such objects have been earlier identified as domesticated, because it was thought that the torn area derives from threshing, which is not necessarily the case. An additional problem in relation with Tathrachis internodes is that although diagnostic for domesticated species, they also occur as mutations with up to 10% in wild populations. When investigating early einkorn finds, we also need to be aware that early reports may be biased, as is indicated by the changes made to the identification of einkorn from the Valichori visible in the table. Throughout the years, einkorn identification at the site changed from mostly domesticated to mostly wild, mostly domesticated again, and back to mostly wild. As discussed before, it is difficult to differentiate between grains of wild and domesticated cereals. There may be even problems to distinguish between different genera, as for example is the case for wild einkorn and wild rye. We will consider now the phylogeny of the wheat species. As the two schemes show, there are a number of simplified and more complex outlines of wheat phylogeny. To date, 2 wild and 1 domesticated form of the diploid einkorn group are known. The 2 wild are Triticum urartu and Triticum boeoticum. The domesticated is Triticum monococcum. The term einkorn is somewhat misleading because the species is not generally one-grained. The wild Triticum urartu is 2-grained and is the donator of the AA genome in the tetraploid turgitum group. Triticum boeoticum occurs in 3 genetic varieties, alpha, beta, gamma, with beta representing the variety important for einkorn domestication. This is the variety Taudar. The Taudar variety also holds 2 grains per spikelet, while the Aegilopoides variety usually holds 1 grain per spikelet. Triticum urartu is genetically easy to separate from Triticum monococcum. However, morphologically, it is indistinguishable from the 2-grained Triticum boeoticum varieties. The picture shows a study by Kilian and colleagues, who measured genetic distance between 350 wild einkorn specimen and 84 domesticated Triticum monococcum specimen. The 3 races of wild einkorn, Triticum boeoticum, alpha, beta, gamma, are shown on the left. Only race beta has been domesticated, grouping together with domesticated einkorn. Triticum urartu, colored in green, in contrast is genetically clearly distinct and has not contributed to the modern genome of domesticated einkorn. Wild einkorn variety B has so far only been discovered in the Karacadag and the Karadag mountainous region in southeastern Anatolia, shown as light blue dots in the map inlet close to the modern towns of Sanliurfa and Diyarbakir. Famous archaeological sites such as Nevadicori and Chayöni are located in this area. Earliest archaeobotanical finds of domesticated einkorn come from further south, from Tel Muraybet in Syria, but from this region no modern specimens have been analyzed. Here are the distribution areas of both wild forms. The quite northern distribution area of Triticum urartu in red underlines the generally underestimated role of the Transcaucasus in the evolution of cereal crops. Triticum boeticum has a better reproduction rate than Triticum urartu under varying ecological conditions, which may explain the fact that domesticated einkorn evolved from Triticum boeticum. Looking at the oldest einkorn finds, we see that the two-grained forms are early in the Middle Euphrates region, while the one-grained forms appear later. It is unknown, however, if Triticum urartu or the two-grained Triticum boeticum variety Taudar is represented here. Although there is no direct cultivar of Triticum urartu, it is likely that early Epipaleolithic and PBN sites contain this species. Direct proof can only happen once aDNA analysis is able to extract sufficiently long sequences from carbonized plant remains. When looking at the regional distribution of einkorn finds, we recognize that indeed it is mostly present in the northern Fertile Crescent. In the southern Levant, this morphotype hardly occurs. Theoretically, the comparison of genotypes in modern crop varieties and wild populations allows identification of the wild population which is most similar to the crop species, that is, which may be considered the progenitor of this crop. The main problem is that we don't know how many of the early wild and original cultivars are still extant. In the recent past, two different views in relation with the genetic results existed. Hoyne advanced the view that crops are monophyletic and that domestication evolved fast under selection through Paleolithic humans. Brown, Ellaby, Özkan, Kilian and Wilcox held the view of a polyphyletic origin of crops without goal-oriented selection during the last Pleistocene and early Holocene. They thought that continuous hybridization of first domesticates with wild species slowed the domestication process down. We know, meanwhile, that both are correct. While most archaeobotanical evidence supports the development of domesticated species in different areas of the Fertile Crescent, einkorn may have indeed been domesticated in only a narrow region in southern Turkey. The duration of the whole process, including the full cereal assemblage, must have developed over a long period of time, roughly over several thousands of years, a perspective that has been named as the protracted domestication. Let's turn now from the diploid to the tetraploid wheat species, which occur as hulled and free-threshing subspecies. The table provides an overview on them, and all go back to wild emmer wheat. Another important species that is a genome donator to the tetraploid wheats is Aegilops, goat grass. The figure on the left shows the genetic relation between different tetraploid wheat species. The eastern wild emmer seems to be closer related with the domesticated species than the western wild emmer. It is again the Karajadag region where the wild species appears that is closest related to domesticated emmer. An unsolved question is why there are no traces of southwestern wild emmer races in modern domesticated populations. Archaeobotanical data also suggests that emmer wheat has been domesticated at two different locations in the southern Levant and in southeastern Anatolia. This fact is also reflected in the archaeobotanical findings, which are equally rich in the northern and in the southern Fertile Crescent. The picture shows percentages of emmer and einkorn spikelets from different sites in the Fertile Crescent. Domestic types of emmer are present from about 10,500 BP, however, they become dominant only around 9,300 BP. The timing of the domestication process is a complex issue, as some of the later sites, such as El Kerk, interrupt an assumed continuous increase in domesticated types. Such opposed trends must be expected in relation with environmental change and are also recognized in later periods, such as at Chalcolithic Tel Tawila in Syria. The archaeobotanical evidence indicates that it took roughly 2,000 years until domesticated forms became dominant in the plant assemblages. Non-climatic reasons for such a slow development have been more frequently described than climatic ones. These comprise the harvesting of unripe ears to avoid seed dispersal in wild ears, which also hindered selection towards indehiscent ears. Other behavioral reasons could be collecting spikelets from the ground, which increased the amount of wild grains in the grain storage that was partially sown later, or just continuous gathering, which would result in a permanent mixing of cultivated wild and domesticated forms. Having a look at the second genome of the tetraploid weeds, our archaeobotanical knowledge becomes more vague. This is the AAGG genome belonging to the Triticum timofevi group. Domesticated timofevi wheat, also named Sanduri wheat, is today an endemic species located in a narrow region in western Georgia. Its wild progenitor, however, has a much wider distribution. Triticum araraticum, the wild progenitor of timofevi wheat, is today spread over the Transcaucasus and the whole northern and eastern Fertile Crescent, overlapping there with Triticum dicocoides. Morphological differentiation of these two wild forms is practically impossible. We know today that evolution also comprises dead ends with species that become extinct. Looking at the large distribution area of both wild species, the question arises why Triticum dicocoides was more successful in terms of the widely distributed cultivar emmer. This is of course a modern perspective, and we don't know which role the species had in the past. Jones and Valamotti were the first to discover spikelet forks that did not resemble the well-known hulled weeds emmer and einkorn. The new type has been found at the Aegean site of Aserostumba and later as well in other sites. According to the authors, all three forms were cultivated together within one field. This would mean that ancient farmers did not differentiate between these species, at least not to a degree to consider their separate cultivation. Considering the ear morphology, particularly the two tetraploid species, emmer and Timofefi wheat, are very similar. While the wild forms Triticum dicocoides and Triticum araraticum are morphologically almost indistinguishable, the domesticated types Triticum dicocum and Triticum timofefi show a distinct difference in their spikelet morphology. Therefore, many archaeobotanists consider the third gloomweed type to represent Triticum timofefi, but only recently this view was supported by DNA analysis. We will hear later about this in a presentation. The development of hexaploid wheat probably took place more or less simultaneously with the tetraploid forms. There are no hexaploid wild forms, and the domesticated species evolved from hybridization of tetraploid wheats with goat grass, namely Aegilops squarosa or synonymous Aegilops tauschii, which provided the DD genome. Hybridization of Aegilops with tetraploid hulled wheat led to spelt, Triticum spelta or Triticum aestivum subspecies spelta. Spelt developed into the hexaploid naked wheat through mutation. Both spelt and the hexaploid free threshing wheat have the same genome formula. The map on the left shows the area of the modern natural distribution of Aegilops squarosa in hatched gray. It is an Irano-Turanian steppe plant with a large climatic amplitude. The black dots indicate what has been earlier considered as secondary habitats. Recent genetic investigation, however, suggests that the ancient area of distribution of Aegilops squarosa was extending as far as modern-day Syria, and that the black dots in the map rather represent relic habitats of a formerly wider distribution of the species. Barley belongs to the same tribe as Triticum, the Triticeae in the family of the Poaceae. All forms of barley are diploid. The classification comprises two-row and multi-row types. In the literature, you will find various species names, which may sometimes be confusing. The traditional classification uses the 4 species names you see in the table, whereas the modern classification groups the wild, the domesticated 2- and 6-row forms under the species Hordeum vulgare and provides individual subspecies names. A brittle form of 6-row barley, Agriokriton, has meanwhile been identified to represent a secondary hybrid of the wild Spontaneum and the domesticated 6-row barley. The picture shows grains and rachis internodes of wild barley on the left and of domesticated 2-row barley on the right. As we have seen for wild and domesticated emmer in one of the previous slides, differentiation of the wild and domesticated barley is straightforward when considering the attachment scar on the rachis, with smooth scars in the wild and sharply edged scars in the domesticated form. As you can see in this and the following slide, barley is found in almost all sides of the Fertile Crescent. We can consider it as the most widespread cereal crop in the Near East throughout all periods. Wild barley has a very broad ecological amplitude, with a much wider distribution area than the wild hulled wheat species. Wild barley is particularly frequent in Near Eastern oak park woodland communities and grows to a precipitation limit of 250 millimeters. Barley has early been carried to Cyprus, where the wild progenitor is not native. There are clear genetic differences between eastern and western wild barley, which is marked on the picture on the right. It is assumed that these allelic differences are ancient. Two-row barley, Hordeum distichum, evolved directly from wild barley through selection, resulting in a tough rachis. Multi-row barley, Hordeum vulgare, emerged through selection of mutants with fertile lateral spikelets. In both of these forms, Hordeum distichum and Hordeum vulgare, there are hulled and free-threshing varieties. Securely identified domesticated two-row barley has been found from the PPNB onwards. As with emmer wheat, the domestication process of barley took at least 2,000 years. The question on independent domestication events led Morrell and Clegg to genetically investigate wild barley from 45 different locations. The genetic composition shows that the largest genetic difference exists between the Fertile Crescent and the region east of the Zagros Mountains. The pattern shows that it must have been domesticated at least twice. First, in the Near East, probably in the western part of the Fertile Crescent during the PPNB, and second, 1,500 to 3,000 kilometers further east, probably at the eastern fringe of the Iranian plateau. The molecular dating of this second domestication was set 2,000 years later than in the Fertile Crescent. There is an ongoing debate on whether the second domestication goes back to some kind of knowledge transfer from the west to the east, or whether it took place independently. Let's now turn to the domestication of pulse crops or legumes. Pulses belong to the most valuable food resources due to their high protein content of 20 to 27% and a high amount of essential amino acids like lysine and tryptophan. Furthermore, their root nodules contain rhizobium bacteria that are nitrogen binding and thus can improve the fertility of soils. Pulses generally occur with lower abundance and frequency in archaeological sites, which is not only for economic or nutritional reasons, but also related to processing methods that differ from those of cereals. The crop species among the pulses are straightforward in identification based on their species' characteristic shapes and sizes. They share the typical characteristics of domestication with the cereals, but their wild and domesticated forms are harder to differentiate. Lentil is cultivated globally today. The natural distribution of the wild progenitor extends into east of the Fertile Crescent, reaching also northern Iran. The cultivar, genetically most similar to wild lentil, is located in southeastern Anatolia and northern Syria. The low genetic variability of modern cultivars suggests a single domestication event. Habitats of the wild progenitor are the same as for wild cereals, mostly on stony ground in steppe environments. The pods are small and usually contain only 2 seeds. Therefore, yields are low, between 500 to 1,500 kg per hectare. Seed sizes of wild lentil range between 2 to 3 millimeters. In early records, sizes of wild and domesticated lentil overlap, making a differentiation in most cases impossible because the shapes are identical in the wild and the domesticated form. A general increase of seed sizes only appears with the fully developed Neolithic period. Lentil appears in larger amounts since the Epipaleolithic, usually together with wild cereals. From the PPNB onwards, it is represented regularly in archaeological sites. Garden pea belongs together with lentil to the oldest domesticated pulses. The wild forms of Pisum sativum can be separated into two morphological types: Pisum sativum subspecies elatius, a climbing plant with Mediterranean distribution, and Pisum sativum subspecies humile, a low-growing plant with distribution in the Fertile Crescent. Humile populations from Israel show the best genetic similarity with domesticated pea, so that it has been suggested that initial cultivation goes back to this region. Seed sizes of wild pea range between 3 to 4 millimeters. Archaeobotanical specimens can be much smaller. Domesticated seeds are generally larger, but there is a considerable size variability in the ancient records. Pea is found regularly from the PPNB onward. These are usually small transitional forms, which are sometimes interpreted as domesticates. However, clear domesticates should be larger than 4 mm. They appear with the fully developed Neolithic from 6500 BC. Cultivation of chickpea is impossible in Central Europe for climatic reasons. Although it tolerates low winter temperatures, it requires high temperatures in summer. Modern distribution of the wild form, Ciza arietinum subspecies reticulatum, is restricted to southeastern Anatolia. It appears in small stands. The original area probably extended further south. Here a list of the earliest finds of chickpea. They are more or less concentrated in the northern Fertile Crescent. The wild progenitor of bitterwedge is unknown, and generally details on this ancient crop are almost absent, which is probably because it is not cultivated anymore today. It has a strong bitterness that must be removed through extended leaching. In the Mediterranean and the Near East, it has been in use until Roman times. It is found since the Epipaleolithic and probably has been domesticated in the PPNB. Therefore, some classify it as one of the founder crops. In modern crop production, broad bean plays a similar role as lentil, but has been more important before globalization of agriculture and saturation of the market with soybean. The species has a broad ecological amplitude and grows in somewhat dry Mediterranean climate as it does in moderate climates of the higher latitudes. There are three interfertile cultivars. The first is a small-seeded variety, the so-called minor variety, which produces seeds in a length of about 10 mm. A second small-seeded variety, Pauci, is mostly cultivated in India, and a large-seeded variety, the variety major, with seeds of 15 to 20 mm in length. The large-seeded varieties developed late, probably not before 1000 AD. And all archaeological finds until Roman times belong to the variety minor. The wild progenitor is still unknown and may even be extinct. Archaeobotanical finds of pulses are represented differently in the early sites, with lentil being most frequent in the southern Levant. This is also true for the other regions. In the north, bitterwedge is also very frequent. Linseed is also one of the founder crops in the Near East. In European prehistory, the most important oil plants are linseed and poppy. The fibers of linseed reached importance for the production of linen. Characteristics of domestication in linseed are closed capsules and an increase of the oil content in seeds. For the use of the fibers, tall and straight plants are required. The area of distribution is very broad, with primary habitats providing moist conditions. Genetic analysis suggests that linseed was only domesticated once. As already discussed in the session on archaeobotanical basics, seeds of linum are generally underrepresented due to a high oil content, which is also reflected in the table of early archaeological finds. The oldest seed finds are from Epipaleolithic Abu Huraira. Woven textiles are already present in PPNB Nach al-Hemar in Israel. We consider now the domestication of woody species. Apart from the fact that most of the fruits can be eaten in their wild form, domestication of woody species requires artificial change of the reproduction biology of the plants through vegetative propagation. In agricultural societies, this is considered to be necessary because most wild fruiting trees are allogamous. This means that they are highly variable in terms of the taste of the fruits. Unwanted characteristics of the fruits are avoided through vegetative reproduction. Woody species develop fruits only after 3 to 8 years of fostering. Therefore, they are also classified as long-term investment crops. The beginnings of fruit tree cultivation are far from being fully understood. Some species have been taken in cultivation in the Near East. Others, such as grape, may have been domesticated in the Caucasus already in the 5th millennium BC. Many of the Rosaceae, such as apple or peach, originate from Central and East Asia, where this plant family has its area of highest genetic diversity. In general, remains of vegetatively reproduced woody species appear several millennia after the domestication of cereals and pulses, that is, with the end of the fully developed Neolithic and the Chalcolithic around 5000 to 4500 BC in the eastern Mediterranean. This slide shows you some easy methods of vegetative propagation. Grape is, with its cultivation on 9 million hectares of land in more than 60 countries, amongst the most important woody crop plants. Together with olive, it belongs to the earliest domesticated fruit species of the Mediterranean. Grapes for wine production and storable raisins were important trade goods since the Mediterranean Bronze Age. There are more than 10,000 different races today, but only one wild form in Europe and West Asia. The wild fruits have been eaten already from the Middle Paleolithic onward. Differentiation of wild and cultivated species in the seed record is usually based on morphometric studies. For the proof of early wine production, the archaeological records, such as wine presses and other installations, are important. Wild forms, such as those from Upper Paleolithic or Hallo II, have been gathered long before they have been taken into cultivation. Although most of the cultivar finds since the Chalcolithic derive from Near Eastern sites, it is assumed that they have been imported from the Transcaucasus region, where grape has been taken into domestication from around 5000 BC. From there it distributed fast to southern Anatolia and into the Levant. Between the 4th and the 3rd millennia BC, it reached Mesopotamia, Egypt, and southern Greece. Recent results suggest that grape was introduced to Central Europe probably already with the end of the Bronze Age. A secondary expansion took place with the Romans. Genetic research on grape is blooming and will present more interesting insights into the evolutionary history as a crop in the near future. Olive is sensitive to temperature and particularly affected by frost. Seasonal cold sequences around freezing point result in damage and extended frost in dieback of olive trees. Olive is, however, undemanding in terms of rainfall. On lean calcareous soils, 200 millimeters of average annual precipitation is sufficient for thriving. Olive trees have small growth rates and fructification usually starts only 5 years after planting. Furthermore, labor investment in olive cultivation is considerably high because vegetative propagation requires grafting and first yields are produced 8 to 10 years later. The mean annual yield reaches about 60 kg, with a maximum production in the time span of 60 to 100 years. The annual yields are alternating, that is, after a rich harvest, only few fruits are developed in the following year. Modern cultivars and technologies allow extraction of up to 16 liters of oil from 100 kg of fruits. Today, oil and table olives represent different varieties. Nevertheless, sometimes the same variety is used for multiple purposes. Earliest finds of wild olive occur in Epipaleolithic Ohalo II, but have been taken into cultivation not before the Neolithic period. The wild and domestic subspecies can only be differentiated through morphometric methods. Because archaeological presses from Bronze Age sites do not always allow to distinguish between their functional priorities, that is, whether they were used for wine or olive oil production, an assessment of the economic role of both crops is not always easy. Biomolecular studies on olives suggest a minimum of 11 original populations in the Mediterranean, with a total of 9 independent domestication events. Half of the cultivars result from hybridization of one or multiple ancestry lineages, distributed over the western and eastern Mediterranean regions. The genetic studies suggest also one late center for modern Tunisia, where cultivars were introduced by the Phoenicians from the east, while local domestication of wild forms took place simultaneously. Those results are supported by morphometric analysis of archaeobotanical olive stones.